thank you all for coming. This is a good turnout, and I hope you've been able to join us for some of the earlier parts of our DNA series, and know that there's some more to come still. It's my pleasure to introduce Dan Barouch tonight for his talk, as you can see, Prospects for a Vaccine and Cure for HIV. Dan's a professor of medicine at Harvard Medical School, and as you can see, director for the Center for Virology and Vaccine Research at Beth, Israel. He is also part of the Radcliffe Institute of Mass General, MIT, and Harvard. He's going to talk tonight about on how molecular science has revolutionized the approach to vaccine and drug development. And the goal of the lecture is to describe the current state of the HIV epidemic and the prospects for developing a vaccine and a real cure for this disease. Dan's laboratory has explored a series of novel vaccine technologies, advancing in particular a series of novel adenovirus. I have to read this because I know none of this stuff. I may be an epidemiologist, but I do heart disease. Novel adenovirus vector-based HIV-1 candidates, from concept and design to preclinical testing to phase 1 clinical trials, currently underway both in the US and in South Africa. His laboratory is part of the Bill and Melinda Gates Foundation collaboration for AIDS vaccine discovery, the NIH Center for HIV and AIDS Vaccine Immunology, and as you can see here, the Radcliffe Institute. So please join me in welcoming Dan Barouch.
Thank you very much, Janet. It's a pleasure to be here today. Thanks for coming on this beautiful afternoon. What I'd like to try to do is to give you an overview of a research topic that I've spent a lot of time thinking about over the last 10 years, really, which is on how do we tackle the HIV/AIDS pandemic in a most definitive way, which, as I'll argue, is the development of a vaccine and or a cure for this disease. We currently have many drugs that can control this disease, but currently no method that can either reliably vaccinate against this disease or be a definitive cure. Also, it's a pleasure to be part of the DNA series, and I think that from my talk, I think you'll see how we use molecular science, which of course involves recombinant DNA, to both think about the problem and attack the problem. So I'd like to try to keep this as informal as possible, so feel free to interrupt, raise your hand, or just speak out questions along the way. I know there is time for discussion at the end, but I'm happy for this to be a dialogue for the first half of this session as well. So don't be shy and feel free to interrupt. Also, if you don't understand something or if you want to discuss something further, then feel free to ask at any point in time. I've tried to keep the technical jargon to a minimum in this talk, and I'll try my best to explain some actual scientific concepts of what we're trying to do and the way we think about this in as understandable language for a non-specialist as possible. If I lose anyone, then let me know, and also hopefully things will be brought back together periodically. Okay. So first, I think many people will remember in the 1980s and even early 1990s how HIV was at the centerfold of newspaper headlines on a very frequent basis. Over the last 5-10 years, largely due to the advent of therapies, then HIV has to some extent partially receded from public view. And to some extent, that's also one of the dangers, that it's not imminently in the public mindset as much as it once was. It doesn't mean the problem is less severe, it just means that it's not in the headlines every day. The way, say, for example, the Ebola epidemic was in the headlines. You couldn't have missed it if you were awake for any portion of this past year. But I think it's important to keep in mind the scope of the HIV epidemic to start with. In other words, how much of a problem is it? These are some data graphics that we borrowed from an article in National Geographic that looks at the estimates of fatalities for major epidemics of infectious disease throughout the world, throughout history. So, for example, these are different epidemics that have occurred in the world over time, with the size of the red circle essentially reflective of the number of fatalities of different infectious disease. So you can see at the bottom the SARS epidemic, which was very much in the headlines about 10 years ago, or in 2002, 2003, claimed almost 800 lives, clearly something of substantial magnitude. The Haiti cholera epidemic, starting in 2011, has claimed over 6,000 lives. The Ebola epidemic, these numbers are a bit outdated. I think the current, or the complete counts for the Ebola epidemic, the 2014-2015 Ebola epidemic in West Africa, is about 11,000 people, so substantially larger than the 8,000 here. This was prepared a few months ago, but still about the same size dot, maybe between this size and this size. So a very substantial epidemic that clearly captured the world's attention, huge resources, and a massive global response. The Great Plague of London was 100,000 people. These cholera epidemics were several 100,000 people. The swine flu outbreak in 2009 claimed about 300,000 deaths. But if we keep going up to even the more significant epidemics in history, then you can see that all of these actually are relatively small. So in terms of the scope, say, of the Ebola epidemic, it was actually fairly small compared with some of the other epidemics that you'll see. For example, if we keep going up, and this is just to show a few of these, the plague in around 1900 was about 10 million deaths. The Asian flu in the 1950s was about 2 million deaths. The 1918 flu claimed almost 20 million lives. And just for comparison, the HIV/AIDS epidemic so far has claimed 39 million lives. So really a very substantial epidemic that takes its place in history as one of the defining infectious disease epidemics of all time, superseded only by the Black Death in the Middle Ages, which claimed an estimated 50 million lives, and the plague of Justinian during the Roman Empire that claimed roughly 100 million lives. So that's sort of where the HIV/AIDS epidemic falls in terms of historic proportions. More discreetly, the number of adults and children living with HIV is approximately 35 million currently, and there's approximately 2 million new infections per year. And these numbers over the last couple of years have remained largely constant. So the number of, the mortality rates have started to drop, which is a good thing. But the number of people living with HIV has actually continued to increase over time. To some extent, that's good news. That means that people are actually living longer, but that also means that the total global burden of the disease is growing and not shrinking. In addition, globally, HIV disproportionately affects young women. This is data from South Africa in which you can see that the greatest burden of this disease is in young women, essentially as early as age 15 and going up to the early 30s. And you can see that throughout Africa, really, this is data from South Africa, but this is the same in many other regions in Africa, that the burden of this disease is much larger in young women compared with young men. And currently in South Africa, then in early adulthood, about 35% of women are HIV positive. These are some data in a hyperendemic region that we do some clinical research with, and this is one of our sites for our vaccine trials. These are data from Slim and Quraysh Abdul Karim, recent data showing really still a stunning HIV prevalence and incidence rate in this community. So this is a community outside of Durban, South Africa, in which over the last few years, the HIV prevalence rate really has not declined much at all, despite the introduction of antiretroviral drugs to the region and massive behavioral and educational campaigns. Then essentially those do not seem to impact the transmission rate of HIV in this population. By age 16, about 8% of women are already HIV positive. In this community, by age 20, about 25% of these women are HIV positive. And by age 24, over 40% of these women are HIV positive. So that between the 18 to 35 year time frame, there's about a 9% incidence rate per person per year. So, and this is with the availability of antiretroviral drugs for prevention as well as behavioral interventions. So clearly something more needs to be done. This is also a clinical research site, a site called CAPRISA that we're working with, and it's a challenge doing research in the developing world. But this is actually a very well established clinical research site that has made some of the most seminal advances for both HIV and TB. And so this is a township, and they're associated with the Nelson Mandela School of Medicine. And this is one of our sites for our vaccine and therapeutic trials. So that's globally. Clearly, the vast burden of the disease is global. And the largest percentage of HIV cases is in sub-Saharan Africa. But how about the US? It's still a major problem in the US. In the US, there's about 50,000 new infections per year. And about 2/3 of these, 64%, are in men who have sex with men. Also, there's a certain level of complacency in the US that in young people, the incidence rates are increasing, not decreasing. Particularly in individuals 13 to 24 years old, there's been a 15% increase in HIV incidence rates. And you can see that there's a geographical maldistribution of cases, with the most HIV cases in the US in the South, as well as in the Northeast, including here in Massachusetts, as well as in Washington, D.C. So Massachusetts, New York, and Washington, D.C. areas, as well as the South, is where there's the majority of HIV cases in the US. In fact, some cities, such as in certain areas of downtown DC, have such a high HIV incidence rate, they would actually qualify for US PEPFAR funding if they were in Africa. So people don't realize that in the US, in certain regions, in certain cities, then the incidence rates actually are still very high. Also, I think most people know that the number of the mortality for HIV very thankfully plummeted in the mid-1990s as a result of the introduction of combination antiretroviral therapy, which is, as we talked about, is life-saving but not curative. However, from the late 1990s, so for the last 10 or 15 years, the mortality rate in the US has largely remained constant. So despite continued medical advances, then we haven't made a lot of headway in HIV therapy in the US in the last 10 or 15 years, which is a surprise to many people. And that's not entirely due to the lack of medical interventions. It's due to many societal and social and economic issues associated with this disease. Another thing that is a surprise to most people is that the majority of HIV-infected patients in the US actually are not adequately treated. So these are recent data from the CDC showing that only about 3 in 10 people with HIV in the US are actually receiving suppressive antiretroviral therapy. This is actually a surprise to me as an infectious disease clinician. So I wear 2 hats. I'm trained as an infectious disease and internal medicine trained physician, as well as a basic scientist as an immunologist. And so for me, HIV research is really a merging of my clinical interests and my basic research interests. But despite all my background, I was actually very surprised to see this, that in the US, about 15% of people with HIV are not diagnosed. Of course, if you're not diagnosed, you can't be treated. But only about half of the people who are diagnosed actually are in a stable medical care situation, which really speaks to the socioeconomic side of this disease. And then of those, a small percentage are not prescribed ART, and a small percentage are not adequately viral suppressed. But when you multiply all these things up, it's called the cascade of care. The actual total number of people who are fully suppressed on antiretroviral therapy, it's not 100% like some people think. It's actually about 30%. And of course, the 70% of people in the US who are not adequately treated are the ones who have viral loads that can continue to transmit to other people. And that's one of the reasons why we've made little progress in HIV care in the US in the last 10 years. So how do we end the HIV epidemic? Clearly, antiretroviral therapy is the backbone, and that's absolutely necessary. But we also believe that it's not sufficient, that antiretroviral therapy alone is not sufficient to cure disease. In fact, if you look at history, then therapeutics alone are usually not sufficient to eradicate an infectious pathogen. So what we need to do is we need to find a cure, and we need to find a way to prevent the disease.
I have a question.
Yes?
Can you compare the geography of America with Europe and Asia, what the prevalence is?
So in Western Europe and in the industrialized countries in Asia, it's relatively similar. I don't know the exact numbers because this was really a US-based study, but it's not felt to be hugely better, although there are more social supports in other countries and there is more universal health care in other countries, then there's still a substantial cascade of care. I don't know the exact numbers. I don't know that the studies have actually been done to be quantitatively precise about that. But the concept of a cascade of care, that on each step on the pathway to actually having suppressed virus, then you lose a percentage of people, still holds. Of course, in other areas of Europe and Asia, then it's vastly different. So what I'm going to do for the next 5 minutes or so is I'm going to digress and tell you a little bit about HIV as a virus. So if I lose a few of you, then don't worry. I'll come back to. I'll tie things up in a few minutes. So this is a picture of an actual virus. This is a cell. The pink thing is a cell, and these green blobs are viruses that are budding off the cell. So this is a cartoon of what an HIV virus looks like. It involves a nucleic acid core, an RNA core. So I know we're in a DNA series, but this is RNA. It's similar. It goes through a DNA intermediate, so it still qualifies for this lecture series. It is an RNA virus. There's proteins that surround the RNA that make the viral capsid. Essentially, it's almost the ball that contains the RNA. And then at the very surface is a lipid membrane. It's sort of a detergent membrane, a sort of floppy surface. And stuck in the membrane are the coat proteins, a single protein called HIV envelope, also known as GP120. So don't worry about the names. Just remember that on the surface of the virus, there's a single protein that's called HIV envelope or GP120, and that has to be the target of antibodies. So whether it's antibodies induced by the disease or antibodies induced by a vaccine, then that's the target. If we look at the life cycle of how does HIV actually infect a cell, then it's shown here. This is the virus that I showed you before, a little smaller. And this is, say, one of your cells, if the virus is going to infect you. It enters the cell by attaching to a surface protein on the cell called CD4. You probably have heard that in HIV infection, your CD4 counts go down. What that actually means is that the number of CD4 T cells in your body declines. This is a CD4 T cell. And it's critical because CD4 is the molecule on the cell that allows the virus to go in. Okay? So once the virus enters, then the RNA genome is reverse transcribed into a DNA template. So basically a carbon copy, almost a photocopy of the RNA is made as DNA. This uses a viral enzyme called reverse transcriptase. And this is something unique to this class of viruses. So DNA is then made from the RNA, and the DNA goes into the nucleus and integrates into your host chromosomes. That's a critical event in the HIV's life cycle, because that event of integration of the viral DNA into your host DNA is the reason why we can't cure HIV. So that's maybe one good take-home lesson from this lecture, is that is the single most important reason why we cannot cure HIV infection. It's because the virus integrates into your host chromosomes, and then a subset of those cells essentially the virus goes to sleep. If the virus is not actively producing viral proteins, it can't be targeted by drugs. And therefore, the cell essentially lives on for years. And if you stop therapy, if you're HIV infected and you stop therapy, the virus starts to replicate in those cells again, and the virus comes back. So it has the potential of repopulating a whole nother cycle of viral replication. Yes?
I recently heard that a lot of our DNA, the introns, are retrotransposons from other viruses, not necessarily HIV but have been suppressed.
That's true. So what this gentleman was referring to is that in every single person's cells, then there are pieces of retroviruses. In other words, viruses that are cousins of HIV, that presumably viruses have lived on with us for millions of years. And those pieces of viruses don't cause a problem because they're not intact, they're not full viruses. They can't actually reactivate them to make a virus. So they're essentially pieces of DNA that are largely almost irrelevant. They're just sort of sitting there. I mean, in your genome, only a small fraction of the genome actually is genes that are relevant for your cells to live. And so within the cells, a lot of the junk areas contain sequences that reflect portions of genes that look like HIV. And that's just normal part of being human. Yes?
I got a question. In theory, in our wildest dreams, could we use a technique, a gene editing technique like a CRISPR-Cas9 to excise the HIV?
Yeah, absolutely. So that's actually gene therapy is one of the strategies for HIV cure strategies now. And I can get to that maybe towards the end of the talk more. But people are actually targeting, people can do that. It turns out it's probably easier to target the co-receptors such as CCR5 disruptions, which is the actual strategies that people are using. But absolutely that's one of the strategies that people are using. So this integration event is mediated by a viral enzyme called integrase. It's not terribly creative. But again, I'm emphasizing it because this is the molecular event that is the reason why we can't cure the disease. Then the integrated DNA, then as you probably know from previous DNA lectures, transcribes RNA. And the RNA can either translate viral proteins, and the proteins in the viral RNA get packaged at the cell surface, and then new viruses bud off. So one infected cell can have many different outcomes. It can either go to sleep and essentially the virus just stays latent, potentially for months, maybe even years. Or it can actively produce virus, in which case it can produce hundreds, thousands, or millions of virus particles. So the reason why I went over this is because this is critical for therapies. So all of the HIV therapeutic drugs target these very unique aspects of the viral life cycle. So there are currently more anti-HIV, antiviral drugs than antiviral drugs for all of the viruses put together, just to show you the impact and the pharmaceutical effort to develop HIV drugs, which has been one of the biggest pharmaceutical successes of our generation. So initially, a large number of HIV drugs target this reverse transcription step. They're called reverse transcriptase inhibitors. AZT, you may have heard of, is one of those type of drugs. The drug class that really transformed the AIDS epidemic in the mid-1990s were called protease inhibitors that target this protease enzyme. I didn't talk about this much, but it's an enzyme that cleaves this initial protein into the individual structural proteins that make a viral particle infectious. And more recently, integrase inhibitors, which target this step. And so these three steps, some fusion inhibitors and other receptor antagonists, but for the most part, the vast majority of HIV patients who are prescribed antiretroviral drugs are prescribed some combination of reverse transcriptase inhibitors, protease inhibitors, and integrase inhibitors, and this is the backbone of HIV therapy. So in an untreated individual, what happens is that an untreated individual is infected, virus in the first few weeks, shown in red, goes up sky high, and then is controlled partially to a so-called viral set point. The virus usually doesn't kill the host in the first few weeks, and the host can't clear the virus, so they sort of end up in a stalemate, where the host is essentially killing very large number of viral particles per day, and the virus continues to replicate every day. And this is an analogy of a faucet in a bathtub ongoing, but the drain also open. So a lot of water is being poured in, but a lot of water is being drained out at the same time. Eventually, if the individual is not treated, then more than 90% of people, after a period of 8 to 10 years, develop constitutional symptoms, opportunistic infections, and death due to the disease. So there are, there's a substantial benefit to bringing antiretroviral drugs to the developing world, shown here, and it's one of the one of the most life-saving interventions that the Western world has done for the developing world, and is led by the US, as I think people know. That in the absence of antiretroviral therapy, there would have been about 2 to 2.5 million deaths per year, and now it's down to about 1 to 1.5 million deaths per year. So really a substantial number of lives saved by antiretroviral therapy. But it's not perfect. As we talked about limitations of antiretroviral therapy, it doesn't cure the infection. It doesn't eradicate virus. There's problems of compliance, toxicity, drug resistance. There is still inadequate access in developing countries. Only about half the individuals in the developing world who need the drugs are able to take them. And these drugs can also be used for prevention, for preventing HIV. But there's a lot of challenges to actually implement that on a societal level. So let's talk for a few minutes more about HIV vaccines in particular. Is an HIV vaccine necessary? Certainly history is on the side of vaccines to prevent or to rid the world of viral epidemics. And this is an opinion piece by several leaders, including Tony Fauci, who wrote this editorial about a year ago and said, ultimately, we believe the only guarantee of a sustained end of the AIDS pandemic lies in a combination of non-vaccine prevention methods and the development and deployment of a safe and effective vaccine. So clearly, we need a vaccine to end the AIDS epidemic, but until we have one, then we need to implement all of the other non-vaccine prevention methods that we have. So why is there no AIDS vaccine today, despite over 30 years of concerted worldwide research on this subject? It's a good question. I mean, it's not due to lack of trying. It's actually one of the large biomedical research enterprises so far has not been successful. So before trying to answer this question, how did Edward Jenner develop a smallpox vaccine 200 years ago without the benefit of our knowledge of DNA and modern molecular biology and immunology? Clearly, the development of smallpox vaccine was predicated on several aspects of the virus. There was a clear clinical correlative protection for variola virus, which is smallpox. There was limited antigenic diversity, which means that smallpox is a virus, the DNA sequences of the virus from one person to the next person were pretty much the same. So you're dealing with a single virus as opposed to many different strains of the virus. And broad immune responses could be induced by attenuated viruses. So a classic method of developing vaccines is making a weakened version of that doesn't cause the disease, but is sufficient to raise immune responses that can then target the real infection when it arrives. In contrast, the challenges in the development of a prophylactic HIV vaccine are unprecedented in the history of vaccinology. Unlike the smallpox virus, for HIV, there is extensive viral clade and sequence diversity. In fact, the amount of genetic diversity of HIV within a single infected individual is greater than the diversity of global influenza, just for comparison. So if you need a new flu vaccine every year, then how are you going to make a vaccine against not just the strains of virus in one person, but HIV for the world? The second thing is what we talked about already, that HIV integrates into the host genome, so it establishes latent viral reservoirs. Also, not a single person has cleared HIV infection through immune responses. There's been one person who probably has been cured by a series of bone marrow transplantations. But there isn't a single individual that we know about that's cleared HIV from immune response, so we don't know exactly what immune response to induce. The virus can mutate rapidly and evade both antibody and cellular immune responses. And antibody responses can be induced to target that one host protein that I mentioned at the beginning, but typically those antibodies are not broad. They don't target many different viruses. So there's no method that exists to induce broadly reactive neutralizing antibodies. And as we mentioned, attenuated viruses are often used for vaccines for things like smallpox, measles, mumps, rubella that some of you may have had. But for HIV, attenuated viruses are too unsafe. So currently there is no safe way of making an attenuated version of HIV. In addition, there's no small animal model of disease, thus requiring the use of more expensive and cumbersome non-human primates for preclinical vaccine testing. There would need to be some level of animal testing before the FDA would allow clinical trials to be done. And there's also, a reality is there's much less pharmaceutical interest in developing vaccines as there is, say, for developing HIV drugs. Ken Kelly and I were talking about that just before the start of this lecture. So there's many different challenges. I'll just highlight two of them. So this is a schema showing the extensive global HIV genetic diversity. So here you can see in different areas of the world different pie charts, and essentially the different colors on the pie charts reflect different strains of HIV called clades. Don't worry about the specifics. Just notice that there's many different colors all over the place in the world, and that just highlights the diversity of virus strains worldwide. The second problem is the envelope protein. I mentioned that at the beginning. There's a single protein called HIV envelope that's on the surface of the HIV envelope. Again, not terribly creative name, but this is the one and only protein that can be targeted by antibodies to hopefully prevent the disease. So just to illustrate the problems of generating antibodies against this protein is shown here. This is actually a real structure. So this is not fiction. This is not a model. This is an actual real structure of the HIV envelope protein that was solved just a year and a half ago. You can see that these are the different protein chains. The green is the GP120 protein, and the red is the GP41 protein. Imagine that the viral membrane is here, and this here is the external world. So basically, antibodies that you would need to make by vaccination to block this disease would have to come in from a direction such as here or here and essentially bind to some of the green protein chains. This is a space filling model in which you see what the globular protein actually looks like. But the key thing is that the virus has devised a way to almost become invisible to the immune system. And it does this by covering almost its entire coat with sugars, also known as glycans. By covering the protein coat with sugars, then it's much less reactive to antibodies, because most people's antibodies bind to proteins and not sugars. And so a model of what those sugars look like is shown here. So you can see the sugars are shown in blue and are probably floppy like this. And this is about the number and dimension of sugars. So you can see if you're an antibody and you need to bind to the green stuff to actually neutralize the virus, and you're coming in here, then the virus is going to be almost invisible to you. So this is another reason why developing an HIV vaccine is so difficult. So how have we done as a field? In 30 years, there's only been 4 HIV vaccines that have been tested for clinical efficacy. So think about that. In any other field of medicine, cardiology, oncology, even HIV therapeutics, at any point in time, there's tests of dozens of different concepts for activity in human clinical trials. But for HIV vaccine development, there's only been 4 concepts that have been tested for clinical efficacy in over 30 years. So I would argue that that is essentially insufficient for the global magnitude of the epidemic. And it really is an entirely another talk that would last a whole afternoon to really discuss why this is the case. But as you can imagine, it's going to be a combination of scientific challenges, funding, logistic issues, and many things put together is really a barrier for developing more HIV vaccine candidates. I won't go through these, the technical aspects of these 4, but let me just tell you that 3 of these failed, and 1 was a partial success. So, that's not a great track record for 30 years of global research on something, but it does show some hope that at least one of these trials, shown here, with different vaccine immunogen components, did show a 31% protective effect. So it's too low to be licensed, so it's not a clinically relevant, it's not relevant to the populations, but it is a proof of concept that developing an HIV vaccine is possible. And so here is the actual data from this clinical trial. The number of HIV infections is on the y-axis, and the time of the study is on the x-axis. The placebo group is shown in red, and the vaccine group is shown in green. And you can see that early on in the first year of the study, these curves separated and indicative of a vaccine effect. But then, after time, these largely look parallel. So the interpretation is that the vaccine had some early efficacy, and it was lost quickly, which is the parallel lines after that. But it did lead to some insights. A large statistical immunologic analysis, don't worry about the actual numbers here, but after this trial was done, people looked very closely at what types of immune responses were associated with a protective effect, and not surprisingly, it came down to envelope-specific antibodies. So if you made certain types of antibodies against particular regions of the envelope protein, regions that were a bit more exposed than others, that weren't impeded by all those blue dots that I showed you in the previous slide, then you were able to be protected. So where do we go from here? So there is a signal of protection with one candidate HIV vaccine. This efficacy was marginal, transient, insufficient for licensure. And so the emphasis now is really a two-pronged approach to develop improved vaccine strategies, either a mimic of what was done before, which involved a poxvirus vector with a G120 protein. A vector is an attenuated or harmless virus, such as an adenovirus or a poxvirus, that doesn't cause disease itself, but you can stitch in through recombinant DNA pieces of HIV to make it into a vaccine. Or to develop entirely different vaccine platforms that have various benefits. So this is a study that we published, and I'll just mention in a couple slides what we're doing. We're using an adenovirus as an initial vaccine strategy, followed by an envelope protein to specifically boost antibody responses. And in a preclinical study, we've shown that this vaccine, which is here shown in blue, gives about a 50% protection compared with control vaccines shown in black. So this is the number of challenges required to achieve infection in this animal model, and this is the percentage of uninfected individuals. And these data were published just recently. So the vaccines that we're taking forward involve priming with these viral vectors, so a harmless or attenuated virus that's unrelated to HIV. In this case, it's an adenovirus, a virus that causes the common cold, with HIV antigens stitched inside, followed by a boost with different viral vectors combined with soluble proteins. And we're currently finishing phase 2A clinical trials in the US, in East Africa, South Africa, and Southeast Asia to look at safety and dose confirmation and regimen selections. And if things look good, then in the next probably 2 years, we should be able to start efficacy trials together with our academic and industrial partners. But assuming that things go well, then this will actually be only the 5th concept tested in 30 years in the history of the epidemic. Yes?
I don't know why we don't license things which have, is it 31% efficacy, if I understand correctly? And that's about the male circumcision prevention efficacy rate. The malaria vaccine is also far from 100%. It's in the 40, 50%. If it's cheap, maybe it's a matter of cost effectiveness, but if it's cheap, why not use it?
It's a very good point, and so there's been extensive modeling on how effective does an HIV vaccine have to be of public health utility. In general, the public health authorities in various parts of the world say that they would endorse use of an HIV vaccine if it were about 50% effective. And there's everything from cost of goods to incidence rates and public health, basically the cost of a rollout, et cetera. The tipping point would be a vaccine that's about 50% effective. Of course, in the developing world, it's a very different calculus than, say, in the developed world, where you would probably demand a much higher efficacy of a vaccine. But it's a very good point, and mathematicians and public health authorities and governments argue about that all the time. This particular vaccine could not be licensed because it was more of an academic experiment. There wasn't actually a pharmaceutical company behind it. So there was just no logistic way that the vaccine could be brought to market anyway. And so there are practical considerations as well, in addition to, so even if that vaccine were 100% effective, the way it was tested could not actually be licensed because it would have to be transfer to a company who's willing to take it on, and other non-scientific issues as well. So I'm going to close in the last 5 minutes or so just with some thoughts about HIV cure. It's to some extent the final frontier of therapeutics. We can, as long as we capture HIV patients in clinical care and the country has the resources to support it, then we can treat most HIV patients very well to the point where their virus is suppressed and their life expectancy almost approaches an uninfected individual. But they have to be on therapy daily for the rest of their life, because currently there is no way that we can cure this disease. We can just sort of hold it in check. So why do current drugs fail to cure HIV? We talked about it already. It's because HIV integrates into the host genome. HIV establishes a latent reservoir in long-lived CD4 cells during acute infection. Essentially, HIV integrates into the host genome, and a certain number of those cells go to sleep. That's called a latent reservoir. The viral reservoir is not eliminated by antiretroviral drugs because drugs only target virus that is actively replicating. So
Just to clarify, so the HIV is unusual in its dormant phase?
yes,
That part.
most viruses do not integrate and do not go into latency. And that's one of the cardinal features of HIV. Yes?
Why does the virus stay as latent for so long? So you suggested that it was like nine years or so. What is happening in that period?
So it turns out that so HIV is another unique aspect of HIV compared with many other viral infections, is that it actually is an infection of the very precise immune cells that fight infection. So it's an infection of CD4 T cells primarily. CD4 T cells, so human T cells have certain properties. They can be many different states. One state of human T cells is called central memory cells. And those cells, by their nature, stay fairly quiet in lymph nodes for years. The purpose of those cells is a good thing, that if you encounter a pathogen, let's say if you've had smallpox once, then you'll never get it again. And that's because T cells and antibody cells, B cells, retain memory for a lifetime. And that memory is retained through central memory cells. In other words, specific cells that have particular antigen receptors that are able to recognize smallpox virus or other viruses or other bacteria. And central memory cells, they're designed to live for years. So the same cell can live in your lymph nodes for years. And that's a property of the human immune system. So HIV to some extent hijacks that, and the long-lived property of those cells is conferred upon the virus because the virus is part of the cell now, because the viral genome is integrated. So whatever is the natural history of that cell is the history of the virus. So if those cells have a half-life of 5 years, then you can actually do the mathematical calculations that it would take 60 to 80 years for those cells to essentially die off. And that assumes that those cells are not self-renewing, which they are. So the translation is that those cells persist forever, for the lifetime of an individual.
But what brings it out of that latency period?
That's a very good question. It's not completely known. Certainly some stresses do that. So at any point in time, a percentage of these central memory cells get activated. And when a cell gets activated, it starts producing virus, and it can set off a chain reaction of new virus replication. But the precise methods, the precise molecular details of how a cell goes into latency and come out of latency is actually the cutting edge of research. So one strategy for a cure is not a cure at all, but is if you could lock those cells in permanent latency, if you could prevent them from becoming activated, then you don't need drugs. So one consideration for a cure is, what you would think about a cure is finding a way to actually eliminate all those cells. But a second approach to a so-called cure, or at least a functional cure, would be as if you could somehow lock them in latency and prevent that reemergence. So only one person in the world, we think, has actually been cured of HIV. it was an HIV patient with leukemia who had not one but 2 bone marrow transplants from a donor, and the donor was selected to have specific mutations in one of the receptors that resists HIV infection. Unfortunately, that's not translatable. The next 6 cases, those patients all died from complications of the bone marrow transplant. So it's certainly not something that can be done for HIV patients unless they have acute leukemia and need the bone marrow transplant for treatment of leukemia. And even so, it's incredibly rare and incredibly difficult to do.
Was it a different donor for the other 6 cases?
There were different donors, but the same mutations. So it's just that allogeneic bone marrow transplantations naturally carry about a 25% upfront mortality rate. And then we think that this particular person was cured because he actually had a very rough course post-transplant and required a lot of immunosuppression and therapy and reinduction and a second bone marrow transplant. So basically, complete replacement of essentially every single immune cell by a donor. Yes?
Yeah, just to be clear, those subsequent 5 were being treated for some other leukemia, not just for HIV.
Absolutely. So, I mean, no physician would ever do a bone marrow transplant just for HIV. It just wouldn't be ethical. But all those patients required the bone marrow transplant for treatment of leukemia, and they happened to have HIV. So, of course, other cure strategies are being evaluated. So this is a list of strategies that people are pursuing for HIV cure. So it is thought that the earlier you start antiretroviral drugs, the better. But early therapy is probably not sufficient. Animal studies and human studies have shown that. You may have heard in the popular press the story of the Mississippi baby. Starting therapy within hours of infection at birth appeared to cause a long-term remission after therapy was stopped in that baby. But then after a period of about 2 years, then the virus came resurging back, showing that at least one cell was still infected with HIV and was sufficient to cause a full-blown infection. There's drugs being developed that can activate those cells, the latent reservoir cells. People are also using vaccines. I talked about vaccines to prevent infection. People also are using vaccines to boost immune responses in HIV-infected individuals with the idea that if you can increase a person's own immune system, that their immune system might be able to either control or ideally eliminate the virus. There's also various immunomodulators that try to increase the quality of those immune responses. We talked a bit about gene therapy that would essentially either potentially excise the HIV from cells or disrupt the receptors needed to sustain an infection. We talked about bone marrow transplantation. It's the one success in the world in 60 million infections, and it's not something that could ever be expanded. And our own concept, which we helped develop over the last 2 years, is could we actually use the rare but definable broadly neutralizing monoclonal antibodies, could those be developed as drugs to help target HIV? So I told you that it's very difficult to raise broadly neutralizing antibodies by vaccination, but there are very rare individuals that develop these type of antibodies naturally, and those antibody genes can be cloned out and can be developed essentially as a pharmaceutical drug. So this is a schema of an HIV envelope protein. This is the viral membrane. The virus is down here, and this is sort of the external world out here. So there are a small number of neutralizing antibodies that have been cloned out of HIV-infected people that are very, very good and can neutralize the vast majority of viruses very effectively. Now, these are not raised by vaccination. These are very rare B cells in very rare individuals, essentially pulling needles out of haystacks. So I just don't want people to be confused. By vaccination, so far it's impossible to raise this type of antibody. But by molecular techniques, one can pull occasional antibodies out of people, and the best antibodies potentially might be used for pharmaceutical reasons. So we published a study 2 years ago in which we showed that, in a preclinical study in animals, that infusion of these antibodies can be remarkably effective. You can see in animals that have high viral loads, we can suppress virus to undetectable levels within 7 days for a period of time. And in animals that start off with low viral loads, then we fully suppress virus, and that appears to be sustained over time for over a year. Antibodies, monoclonal antibody infusions can also deplete virally infected cells. So this is essentially one measure of the viral reservoir. And you can see with antibody infusion, it doesn't reduce it to 0, but it certainly reduces it substantially compared with uninfected animals. So I'll conclude briefly, and then happy to take more general questions. The HIV epidemic is, I think, unquestionably one of the most important biomedical challenges of our time, both locally in the US and even more importantly, globally in the world. Molecular science and a molecular-based thinking of the pathogen and the immune response has revolutionized the approach to both vaccine and drug development. And certainly novel HIV vaccine and cure strategies are going to be tested in clinical trials over the next few years. I should emphasize that these are aspirational goals, and there is no HIV vaccine and there is no HIV cure that's around the corner. I can assure you that neither one of these is going to be a licensed product in the next few years. But by harnessing modern immunology and virology, we think that we have an unprecedented opportunity to impact not only the HIV epidemic, but this way of molecular thinking will also likely be very effective to develop strategies against other infectious diseases as well as cancers. So I'll stop there. Happy to take more discussion and questions.